Hey there, everybody, Mr. Larson here. So we're now going to be talking about DNA function and basically what DNA is used for. So before we get to what it's used for, we have to understand how it is set up in our cells. Because DNA is such a long, complicated strand, this very long, skinny strand of information, it has to coil up on itself to be more compact and before we create new cells. So DNA is wrapped around with these certain proteins called histones. Here you can see in this image below. So you have all these histones that the DNA wraps around, and it wraps around more histones, wraps around more histones, wraps around. And then once it's wrapped around a bunch of histones, those histones wrap together to create coils. All those coils create what are called supercoils, and all those supercoils are wrapped around again to create this chromosome. So this chromosome, this itty bitty chromosome found in your cells, is basically a compacted version of DNA. So it's this mile-long strand of DNA found in your cells, all compacted into a very small unit in your nucleus. Basically, the example that you can think of is a spool for a thread. So you have a thread in a spool. If you take out the thread is extremely long. It can go for hundreds of feet, but it's compacted in this little tiny package. So DNA, when it is replicated, has to basically separate itself out. So it's going to now create two what are called complementary strands. So you have one strand of DNA. Before the cell wants to duplicate itself, it has to create two complementary strands. So now instead of one strand, we're now going to have two. How it does this is by using what is called DNA polymerase. It is an enzyme that basically separates the nucleotides, separates those bases, separates our A's and our T's from our C's and our G's in order to create these two different strands. You can see here in this image below, or in the image in the presentation, you can see there is one strand here through the center, but then you see here at the replication forks that there are now going to be two different strands. So DNA polymerase basically unzips the DNA and forms two strands going in opposite directions. So the DNA polymerase is basically moving along the strand and bringing in all these different bases that correspond to the DNA strand. So now you're going to have two exact replicas of that once single strand of DNA. So now instead of one, we have two. So think of it like going to the library. I know people don't go to the library as much anymore. Basically, we use Google. But how do you use the information from Google? So instead of basically taking it off of Google, you have to basically copy it. This is basically what a DNA polymerase is doing, similar as what you would with library books, encyclopedias. You're not allowed to take it home with you, but what you can do is you can create a copy of it. So now there is a copy of the information that's going to be shared with other cells. So basically that is how DNA replicates and why DNA replicates, so that when it creates another cell, it is the exact same information and there is no difference between the two. I hope this is helpful, hope this is useful, and we're going to talk about the next process in DNA replication.